morning to all of you. So today in the practical class we'll learn about how can we use the technique of paper chromatography. So as we know that chromatography stands for separation of compounds from mixtures. So chro means color and graphy means to visualize or to understand the observations. So here in this particular technique what happens, we take a base material as paper. So here we have in any chromatography technique, as I've already learned in a theory classes, we have two types of phases. One is called the mobile phase and another is called the stationary phase. What happens is that if we have to separate different compounds from a mixture, then we have to understand how the different compounds of a mixture interact with the stationary phase and with the mobile phase. So here, as we know that, so chromatography is a method that is used to separate or to segregate different kinds of mixtures into the respective components, and the main purpose is four purpose, as you can see from here: analyze, identify, purify and quantify. Now the major important point of this technique is that we have two types of phases. One is called the stationary phase, which means it is stationary, it doesn't move, and it's a mobile phase. There is a liquid or a solvent with which the particular components will interact, and based on the polarity, based on the solubility, and based on the particular capillary action mechanism, what will happen is that the mixtures will move in towards the solvent with regard to the solvent front. Now, paper chromatography, in most importantly, as you can say, it's a type of partition chromatography. Partition means segregation chromatography, which means, suppose, let's say, let's take a diagram, small diagram, you can understand. So let's take this particular paper strip. Okay, so suppose we have kept this in a particular solvent system. Okay, and let's say we have a different color. Let's say we have a different ink color we take. So this is the solvent front. Okay, now we load the particular samples here. Let's say it's A, it's B, it's C. Now if the samples interact with the solvent phase in a very high affinity, then it will move very faster along with the solvent front. So our stationary phase is cellulose. Okay, and most importantly, our mobile phase is a solvent. Let's say distilled water or any kind of solvent that we want to take. Now, if the particular compounds are having high affinity with this stationary phase, what will happen? The rate of migration with respect to solvent front or solvent will be less. So this particular migration of the solvent is known as solvent front. That is how much solvent has migrated with respect to your stationary phase. So therefore, as you can see from here, cellulose is a particular grade filter paper. So here it's a Wattman filter paper number 1. So we use grade 1 Wattman filter paper in order to analyze and identify the particular compounds as with respect to our mobile phase. Okay. Now as you can see from here, this is a mixture and this will separate into the particular components, and we have this 4 aims of our experiment. Okay. Now, as I've already discussed, now why we tell as differential migration? Now, differential migration means that there is a difference between the affinities of stationary phase and mobile phase, which means if suppose the compounds in the mixture, they are having high affinity towards the stationary phase, then they will have better interaction with the cellulose, and therefore they will not be able to migrate properly with respect to the solvent. And the opposite will occur when we have compounds from mixtures that will have higher affinity towards the mobile phase. So that means it depends on 3 factors. This is the molecular size, polarity and solubility. So these are the 3 particular you can say these are the particular 3 things where the factors which basically influence the migration and the ability of the compounds to be separated from the mixtures. Now, as you can see from here, this is the working principle. It is simple. So we suppose we have 3 types of mixtures. One is like, as you can see, let's take 1, 2 and 3. See, this is the first compound, let's say second and third. Once we separate, see, this one has the highest affinity towards the stationary phase. So it has not migrated at all. Okay, but as you can see from here, this one, it has the lowest affinity with respect to mobile phase, but highest affinity with respect to stationary phase. Which means if I write in a simple terms, affinity if high towards mobile phase, if it's okay, then high migration. Okay, then high migration. Here, high migration. But if we have the opposite case, so high migration means this one is your high migration. When we have affinity high towards the mobile phase, and the opposite will occur when the affinity will towards the stationary phase, as like this. As you can see from here, this one is your having higher affinity with the mobile phase. Okay. Now as you can see from this is the transition slide. This is the sample. So what we have done is we have loaded the sample in the baseline. So if I change the color, suppose this is the baseline. Okay. So this is the baseline basically. This is called baseline. Okay. Baseline. Baseline means the line at which we load the samples at equal distance or equidistant loading. Okay. Then you see this is solvent front. Solvent is there. As you can see from here, it will slowly migrate towards the distance. So if we consider this one, this has the lowest affinity okay, towards mobile phase. Okay, if I consider this one, lowest affinity towards mobile phase, but this one has the highest affinity towards mobile phase. Highest, I am writing highest. Why? Because the more the affinity towards the mobile phase, the better the migration efficiency. Yeah. Now this is the RF. RF is called the reduction factor. It means distance traveled by the component. Component means this one. So this is your A and this is your B. So RF means the calculation of A. So RF, if it's high, so it will be high migration. Okay, that means we have higher affinity towards the mobile phase. Clear? Higher affinity. But if my RF reduction factor is low, then we have low mobility. Low mobility. Here we have low mobility. Here that means no mobility means this will be moving in a very less distance. That is called the RF factor. And the type of paper chromatography, where we have one is called the ascending paper chromatography, where the solvent front moves from downward to the upward direction. This is basically based upon the principle of capillary action. Okay. Capillary action. Then we have descending paper chromatography, where basically this is a clip is there. What we do is we clip the particular paper strip, and the movement comes towards due to the gravitational pull. So the movement occurs due to gravitational pull. Which means that suppose we are loading the samples in the upward direction here, so the sample will come in the action of gravity. So solvent front and the distance migrated by the solute will be based upon the gravitational pull. This is called the radial paper chromatography. Here what happens, we load the samples here and then it moves in the radial directions. It spreads out. Why? Because of capillary action. So the more the migration, the better the affinity towards the mobile phase. The less the migration, the lesser the affinity towards the mobile phase. And like this particular technique is very important when we want to resolve, let's say, amino acids, okay, which has basically very close RFs or retardation factor values, which means when they are very difficult to separate. What happens is that first we do an ascending chromatography, okay, Then what we do, we turn the filter paper at 90 degree, and then what happens? Again we do a chromatography. Now this ensures that two steps of differential migration will occur, due to which we can actually get the real distances or real RFs, rather than having a confusion that which is migrated to what direction. Okay, clear? And basically we have lots of applications in the food industries, in pharmaceuticals, in textiles, and in chemicals. So today we will be doing with the two types of chromatography. So first, in our practical, we will be doing the circular chromatography or the radial one, and then we will be doing the paper strip chromatography. And some of the aspects and limitations are given here. Especially paper chromatography comes with low cost, but the precision technique is limited, so we go for higher order chromatography like HPLC, like GC, and others which have already been discussed in your theory classes. And therefore we can use this technique in only for a qualitative analysis. We try to semi-quantify it using RF factor, but it's basically a qualitative analysis. Okay, thank you so much. Chromatography, one is the paper strip and another is called the radial chromatography or the circular chromatography. In this kind of paper strip, okay, what we will do is we will mark a 2 centimeter line. So from the base, like in this region where the straight path starts, we will first mark it with scale. Okay, not exactly, but approximately it should be straight line. So we mark it here. Why we mark it? Because we need to create a baseline, as I told you. In any chromatography technique, you have to have a baseline, especially for the paper chromatography. So what to do is, we take two sketch pens. Let's say I have red and blue. You will use your own, and I'll mark it here, and I'll mark it here. Here. Now, you have to ensure this, that you have to mark in such a way that your dots are almost at equal size. This is just for showing you, but your dot should be of the equal size. Okay. Now you have to dip this particular filter paper in water in such a way that the filter paper doesn't get like wet completely. Only the basal part, this part, should be wetted. Why? Because so that the capillary action will go gradually. So if you pour more water, then the entire filter paper will get wet. So there's no use of doing the capillary action. So what we will do? We will take this bottle. I'll pour very little amount of water. Okay, this is my water. Okay, so I think this is too much. I'll decrease the water amount. Now it will depend. You have to understand. Okay, I'm just showing you, and you have to place in such a way that it should not fall off inside the water. So I'll decrease more water, and you have to be very careful in placing this part. You just have to place this. See, just place this. Dip with the छोड़ना. Even if you see, it has to be straight. So normally it should go up. It's moving down because you have to be very careful about it. I'm just showing you. You have to do it in such a way that your movement should go upwards. That means you should not dip it completely. I'm showing you why, because most of the people will do it wrong. So you have to dip only the tip part. See, it has already moved. So it should move upwards. Why it move downwards? Because if you are dipping it too much, then it may move downwards. So you have to be very careful while dipping it. That means you have to decrease the amount more. You take very little amount of water in only to dip the tip, and then the water will move up. Clear? Clear what I'm saying? Now, based on your migration, like let's say up to this is migrated. So how will you calculate? First, you will make the things dry, and you will simply take from the baseline. Baseline means from this line, how much distance is migrated by the solvent. That is the first factor. Second factor is how much the solvent is migrated. Simple. And then RF means how much distance traveled by the solute divided by solvent front. Clear? This is the way by which you will calculate the RF of the samples. This is the first point. Okay. The second point is, how will you be able to calculate the radial chromatography? So for that, what you do? So this you have to cut, and you have to make a hole like this, so that the support comes here. So this is a petri plate. Okay, what to do is very simple. First, you move the support Okay, everybody. Support is moved. Now what you do is, suppose you just mark along the circle in such a way that they are equidistant. For showing purpose, I am only showing you. You have to be very careful, because if you are not equidistant enough, then the samples will migrate randomly. They should be a random thing. So you have to do is, first you make a circle, like a small circle with a compass here in this particular. That means what? You are creating a baseline, right? So in that baseline, you create a circle, and then what you do, simply you mark here 1, suppose then 2, suppose let's say 3, okay, and then 4. Now you will have to be very careful while marking.